Hello, my name is Larsa Hannah, and I am doing my disease project on Pseudomonas aeruginosa. Its etiologic agent is any poorly cleaned healthcare equipment, like a physiotherapy pool that has not been refreshed with new water. It can be in respiratory devices, disinfectants, sinks, and cleaning solutions. It is found in moist or water-containing equipment, and mostly any medical equipment that has come in contact with an infected patient's fluids. It accounts for 10% of all hospital-acquired infections. Its reservoir is any uncleaned water, soil, and vegetation. It can be found in a healthy person's skin, ears, and respiratory tract. It favors moist environments. It can be found in vegetables, fruits, or other plants, like flowers that have been absorbing contaminated water. And again, in general, it is found in pools and large bodies of water. The morphology and features of Pseudomonas aeruginosa is that it is a gram-negative bacteria, it is rod or bacillus-shaped, it is motile, it is an opportunistic pathogen, so it won't really cause an infection until the target cell that it is trying to infect gets weakened. It can oxidize glucose and xylose, but it cannot ferment lactose or other carbohydrates. It is a facultative anaerobe, so it can thrive in aerobic, which is oxygenated, or anaerobic, which is non-oxygen, environments. Virulence factors and mechanisms of infection for Pseudomonas aeruginosa is that it requires only a minimal source of carbon, and it thrives in moist environments, and it can actually survive under somewhat high pressures that would eliminate competing flora. It contains pilus, which allows for conjugation of bacterial information, and a flagella, which allows it to move or swim. It has alkaline protease, which lets it live in alkaline environments. It is a cytotoxin, so it is toxic to most other cells. It will infect the cells through the flagellar pili and lipoproteins. And it can also infect through type 2 secretion system, which allows for cytoplasmic proteins to be transported across the cell membrane, and these proteins can send signals to other nearby cells, which aids in the promotion of infections. It grows well at 97 degrees Fahrenheit, which is 36 degrees Celsius, and 107 degrees Fahrenheit, which is 42 degrees Celsius. The diseases caused by Pseudomonas aeruginosa are mostly infections. These infections can occur in pretty much any region of the body, and it can cause swimmer's ear infection, which is also called otitis externa, which is an infection of the external ear. It can cause eye infections like keratitis and conjunctivitis. It can cause pneumonia, urinary tract infections, and in patients with already weakened immune systems, it can cause endocarditis, which is inflammation of the heart valves, and septicemia, which is blood poisoning. And in very rare cases, it can actually cause meningitis too. In the image shown, it is an example of keratitis infection caused by Pseudomonas aeruginosa. So basically, that is an infection of the cornea, and as shown in the image, the patient's cornea is actually being inhabited by Pseudomonas aeruginosa, and their cornea is being eaten away. So if it is not treated as soon as the infection begins, they will go blind in that affected eye. It accounts for 0.4% of hospitalizations, and it causes 10 to 20% of infections in hospitals. The signs and symptoms of Pseudomonas aeruginosa really depend on what area has been affected by it, but if it is the respiratory tract that has been affected, it will cause pain and difficulty when breathing, wet cough, which may have a green or yellow mucus, and the mucus can actually contain blood too. It will cause fever, chills, and tiredness or fatigue. It will cause inflammation or redness of the eyes if the eyes have been affected. In the ears, it will cause pain and liquid discharge, and in more severe cases, it can cause temporary hearing loss, or if untreated, forever hearing loss, permanent. In the skin, it may have rashes and pus-filled pimples. The transmission of Pseudomonas aeruginosa is mostly through food and water, but then again, it is also very commonly transmitted through contaminated hospital supplies, and it is a nosocomial infection. So any medical equipment that has not been cleaned can transmit the bacteria to new patients. It is mostly a waterborne bacteria, so any contaminated water or moist equipment or just anything with water in it or fluids can transmit the bacteria to new patients. The lab diagnosis of Pseudomonas aeruginosa is done when blood or other body fluids are taken from the patient and cultured for the lab test. It is grown on blood agar plates like tech agar, and it is the only gram-negative rod-shaped microorganism that can produce piocyanin. When grown on the tech agar plate, it will appear blue, as shown in the image on the right. It can be grown on eosin methionine blue agar plate, and it tests for the ability to ferment lactose. Now, when it is grown on this plate, it will be able to grow and produce a culture, but it will remain colorless because it lacks the ability to ferment lactose, so it will be colorless, but it will still be able to grow on the agar. It is differentiated from similar species like Pseudomonas florensis through a nitrate test. This test tests for the ability to reduce nitrate to nitrite, and Pseudomonas aeruginosa will appear positive, while Pseudomonas florensis will appear negative on the test. And the image shows how Pseudomonas aeruginosa will appear positive, and it will turn the broth of the nitrate test to a deep red color. It is gradually becoming more difficult to treat due to antibiotic resistance. So, as the bacteria is able to resist antibiotics better, it is becoming more difficult to treat. Vinegar, however, can still aid patients with the swimmer's ear infection, and the antibiotic polymyxicin can be prescribed, along with oral antibiotics like levofloxacin and ciprofloxacin can also be helpful for the patients. Prevention is very important, and most ways to prevent Pseudomonas aeruginosa would really just be cleaning hospital supplies better, sanitizing more, and keeping higher hygiene. Drying the ears after swimming or taking a shower is also very important. Avoiding uncleaned pools and showering with hot water after swimming. Also, the swimsuits and towels or whatever used in the pool should be washed as best as possible. People should avoid wearing eye contacts in the pool and should avoid sleeping with their eye contacts. They should always wear their eye contacts as directed and prescribed. Those who have weaker immune systems, especially patients with cystic fibrosis, should just really try their best to practice high hygiene standards and just avoid swimming as much as they can. Vinegar and ethanol disinfectants have proven to be somewhat effective for aiding the killing of the bacteria. The bacteria immunity is really that there is no vaccine for it. In the lungs, inside the alveolar sacs, there are macrophage cells, and these cells can help the body fight against Pseudomonas aeruginosa. However, as the infection spreads, or really if there's just too much bacteria, the cells will not be able to prevent the infection, and the infection would most likely, in this case, be pneumonia. It is resistant to many antibiotics because it is a gram-negative cell, and the antibiotic resistance has been increasing. So special comments about Pseudomonas aeruginosa is that it was first discovered in 1882 by the French chemist Carol Dizard. It appears as a blue or green color under ultraviolet lighting, and this is due to the pyocyanin and pyoveridin it has. The word Pseudomonas comes from the Greek word false unit, and the word aeruginosa refers to the color blue-green, hence the name Pseudomonas aeruginosa. It has a sweet smell, which has been described as grapes when it is isolated in a pure colony, and it significantly impacts cystic fibrosis patients more than any other patient with a weaker immune system, and far more than patients who are healthy. So here are the references I used when working on this project and finding my information. And I have some multiple choice questions. So question 1 is, which patient is most susceptible to an infection caused by Pseudomonas aeruginosa? The options are a healthy elderly individual, a young female child, a patient with cystic fibrosis, and a healthy adult male swimmer. Question number 2 is, what is one way to differentiate Pseudomonas aeruginosa from similar Pseudomonas species? The options are motility test, nitrate test, gram stain, and mannitol salt agar test. Number 3 is, what is an infection commonly caused by Pseudomonas aeruginosa in hospitals? The options are pneumonia, strep throat, cystic fibrosis, and malaria. So the answers to the questions is here. Question 1, the answer is a patient with cystic fibrosis, as they have a weakened immune system, so they are more susceptible to the infection. Answer number 2 is a nitrate test, and as you may recall, Pseudomonas aeruginosa can be differentiated from similar Pseudomonas species, like Pseudomonas florensis, through the nitrate test, as Pseudomonas aeruginosa is positive and Pseudomonas florensis is negative. Number 3 is pneumonia, and that is because it is transferred through the medical equipment. So hey, thank you very much for watching, and I hope you enjoyed it. Have a great day.